Hello friends and welcome. Today we shall talk about microbial nutrition. The main objectives of today's deliberation are: Number 1. To study the nutritional requirements of microbes. Number 2. To know about nutritional types of microorganisms. Number 3. To understand the mechanism of uptake of nutrients by the microbial cell. And finally, to study the culture requirements of microbes. A nutrient is any chemical required for growth and development. All cells require three things to conduct metabolism: a carbon source, a source of energy, and a source of electrons or hydrogen atoms. The microbial nutrition can be studied under different headings. Number 1. Nutritional requirements of microbes. To obtain energy and construct new cellular components, organisms must have a supply of raw materials or nutrients. Nutrients are the substances that are used in biosynthesis and energy production. The most important of these are compounds containing carbon, oxygen, nitrogen, and/or hydrogen. Microbial growth depends on the metabolism of nutrients and results in the formation of a discrete colony and aggregation of cells arising from a single parent cell. Macronutrients or macroelements. These are required by microorganisms in relatively large amounts. Carbon, oxygen, hydrogen, nitrogen, sulfur, and phosphorus are components of carbohydrates, lipids, proteins, and nucleic acids. The remaining four macroelements, potassium, calcium, magnesium, and iron, exist in the cell as cations. Potassium ion is required for the activity by a number of enzymes, including those involved in protein synthesis. Calcium ions contributes to the heat resistance of bacterial endospores. 15% of spores contains dipicolonic acid and calcium. Magnesium ions serve as a cofactor for many enzymes complexes with ATP and stabilizes ribosomes and cell membranes. Ferrous and ferric ions are part of cytochromes and a cofactor for enzymes and electron-carrying proteins. Micronutrients or trace elements. In addition to the main elements found in microbes, very small amounts of trace elements are required. These are manganese, zinc, cobalt, molybdenum, nickel, and copper. These are normally part of enzymes and cofactors, and they aid in the catalysis of reactions and maintenance of protein structure. Zinc ion is present at the active site of some enzymes but is also involved in the association of regulatory and catalytic subunits in E. coli aspartate carbamyl transferase. Manganese ion aids many enzymes catalyzing the transfer of phosphate groups. Molybdenum ion required for nitrogen fixation. Cobalt ion is a component of vitamin B12. Besides macro and micronutrients, some microorganisms may have particular requirements that reflect the special nature of their morphology or environment. Diatoms need silicic acid to construct their beautiful cell walls of silica. Bacteria growing in saline lakes and oceans depend on the presence of high concentrations of sodium ion. Microorganisms require a balanced mixture of all the aforementioned nutrients for proper growth. Requirements for carbon. Carbon is needed for the skeleton or backbone of all organic molecules, and molecules serving as carbon sources normally also contribute both oxygen and hydrogen atoms. One important carbon source that does not supply hydrogen or energy is carbon dioxide. Autotrophs can use carbon dioxide as their sole or principal source of carbon. Many microorganisms are autotrophic, and most of these carry out photosynthesis and use light as their energy source. Some autotrophs oxidize inorganic molecules and derive energy from electron transfer. Heterotrophs are organisms that use reduced preformed organic molecules as carbon sources. For example, glycolytic pathway produces carbon skeleton for use in biosynthesis and also releases energy as ATP and NADH. Actinomycetes will degrade amyl alcohol, paraffin and even rubber. Burkholderia specia can use over 100 different carbon compounds. Some microorganisms can metabolize even relatively indigestible human-made substances such as pesticides. Indigestible molecules can be oxidized and degraded in the presence of growth-promoting nutrient that is metabolized at the same time, a process called co-metabolism. The products of this breakdown can then be used as nutrients by other microorganisms. Requirements for oxygen. Obligate aerobes require oxygen as the final electron acceptor of the electron transport chain, whereas obligate anaerobes cannot tolerate oxygen and use an electron acceptor other than oxygen. Toxic forms of oxygen are highly reactive and cause a chain of vigorous oxidation. 4 forms of oxygen are toxic. Singlet oxygen. It's a molecular oxygen with electrons that have been boosted to a higher energy state, typically during aerobic metabolism. Phototropic microorganisms often contain pigments called carotenoids that prevent toxicity by removing the excess energy of singlet oxygen. Superoxide radicals. These are formed during the incomplete reduction of oxygen during electron transport in aerobes and during metabolism by anaerobes in the presence of oxygen. They are detoxified by superoxide dismutase. Peroxide anion is a component of hydrogen peroxide which is formed during reactions catalyzed by superoxide dismutase. The enzymes catalase and peroxidase detoxify peroxide anion. Hydroxyl radicals. These result from ionizing radiation and from the incomplete reduction of hydrogen peroxide. Hydroxyl radicals are the most reactive of the 4 toxic forms of oxygen, but because hydrogen peroxide does not accumulate in aerobic cells, the threat of hydroxyl radicals is virtually eliminated in aerobic cells. Not all organisms are either strict aerobes or anaerobes. Facultative anaerobes can maintain life via fermentation or anaerobic respiration, though their metabolic efficiency is often reduced in the absence of oxygen. Aerotolerant anaerobes prefer anaerobic conditions but can tolerate oxygen because they have some form of the enzymes that detoxify oxygen's poisonous forms. Microaerophiles require low levels of oxygen. Capnophiles grow best with high carbon dioxide levels in addition to low oxygen levels. Now, let us try to understand the requirements for nitrogen, phosphorus and sulfur. Nitrogen is needed for the synthesis of amino acids, purines, pyrimidines, some carbohydrates and lipids, enzyme cofactors and other substances. Most phototrophs and many non-photosynthetic microorganisms reduce nitrate to ammonia and incorporate the ammonia in assimilatory nitrate reduction. A variety of bacteria like many cyanobacteria and rhizobium can reduce and assimilate atmospheric nitrogen using the nitrogenase systems. Phosphorus is present in nucleic acids, phospholipids, ATP, several cofactors, some proteins and other cell components. All microorganisms use inorganic phosphate as their phosphorus source and incorporate it directly. E. coli can use both organic and inorganic phosphate. Organophosphates such as hexose-6-phosphate can be taken up directly by transport proteins. Other organophosphates are often hydrolyzed in the periplasm by the enzyme alkaline phosphatase to produce inorganic phosphate, which is then transported across the plasma membrane. When inorganic phosphate is outside the bacterium, it crosses the outer membrane by the use of a porine protein channel. Sulfur is needed for the synthesis of substances like the amino acid cysteine and methionine, some carbohydrates, biotin and thiamine. Most of them use sulfate as a source of sulfur and reduce it by assimilatory sulfate reduction. A few require a reduced form of sulfur such as cysteine. Now coming to the other chemical requirements. Most microorganisms also require small amounts of certain organic chemicals that they cannot synthesize. These are called growth factors. For example, vitamins are growth factors for some microorganisms. Growth factors. Many microorganisms, they have the enzymes and pathways necessary to synthesize all cell components. Many lack one or more enzymes and hence require organic compounds because they are essential cell components or precursors of such components and cannot be synthesized by the organisms are called growth factors. There are three major classes of growth factors. Amino acids. These are needed for protein synthesis. Purines and pyrimidines for nucleic acid synthesis. Vitamins are small organic molecules that usually make up all are part of enzyme cofactors and only very small amounts sustain growth. Knowledge of the specific growth factor requirements of many microorganisms makes possible quantitative growth response assays for a variety of substances. The observation that many microorganisms can synthesize large quantities of vitamins has led to their use in industry. Several water-soluble and fat-soluble vitamins are produced using industrial fermentations. Like riboflavin are produced by Clostridium, Candida, Ashbia, Aromethicium. Coenzyme A by Brevibacterium. Vitamin B12 by Streptomyces, Propionibacterium, Pseudomonas. Vitamin C by Gluconobacter, Erwinia, Corynebacterium. Beta-carotene by Dunaliella. Vitamin D by Saccharomyces. Coming to the second part of microbial nutrition, that is nutritional types of microorganisms. In addition to carbon, hydrogen and oxygen, all organisms require sources of energy and electrons for growth. Carbon sources. Autotrophs. Carbon dioxide is the sole or principal biosynthetic carbon source. Heterotrophs. Reduced preformed organic molecules from other organisms. Energy sources. Phototrophs use light as their energy source. Chemotrophs obtain energy from the oxidation of chemical compounds, either organic or inorganic. Electron sources. Lithotrophs use reduced inorganic substances as their electron source. Organotrophs extract electrons from organic compounds. Four major nutritional classes based on their primary source of carbon, energy and electrons is known. Phototrophic autotrophs or photoautotrophs or photolithoautotrophs. Source of energy is light energy. Source of electrons, inorganic hydrogen or electron. Carbon source, carbon dioxide. Examples include algae, purple and green sulfur bacteria and cyanobacteria. Photoorganotrophic heterotrophy or photoorganoheterotrophy. Source of energy is light energy. Source of electrons, organic hydrogen or electron. Carbon source, organic carbon sources. Carbon dioxide may also be used. Examples, purple and green non-sulfur bacteria, common inhabitants of lakes and streams. Chemolithotropic autotrophs or chemolithoautotrophy. Source of energy is the chemical energy source, inorganic. Source of electrons, inorganic hydrogen or electron donor. Carbon source, carbon dioxide. Examples: sulfur oxidizing bacteria, hydrogen bacteria, nitrifying bacteria, iron oxidizing bacteria. Chemoorganotrophic heterotrophs or chemoorganoheterotrophy. Source of energy, chemical energy source organic, source of electrons, inorganic hydrogen or electron donor, carbon source, organic carbon source. Example: protozoan, fungi, most non-photosynthetic bacteria, including most pathogens. The most common nutritional types are photolithoautotrophs and chemoorganoheterotrophs. Bacteria, Biota rely on inorganic energy sources and organic or sometimes carbon dioxide carbon sources. These microbes are sometimes called mixotrophic because they combine chemolithoautotrophic and heterotrophic metabolic processes. Coming to the third part of microbial nutrition, uptake of nutrients by the microbial cell. Uptake of the required nutrients by the microbial cell is important. Since microorganisms live in nutrient-poor habitats, they must be able to transport nutrients from dilute solutions into the cell against concentration gradient. Finally, they must pass through a selectively permeable plasma membrane. Microorganisms use different transport mechanisms like facilitated diffusion, active transport, and group translocation. Eukaryotic microorganisms do not employ group translocation but take up nutrients by endocytosis. Movement of materials across the plasma membrane is mostly done by two processes. Number 1, passive processes. Substances cross the area from an area of high concentration to an area of low concentration without any expenditure of energy, ATP. Example: simple diffusion, osmosis, and facilitated diffusion. Number 2 is active process. The cell must use energy in the form of ATP to move substances from areas of low concentration to areas of high concentration. Example: group translocation. Passive or simple diffusion, often called diffusion, is the process in which molecules move from a region of higher concentration to one of lower concentration. The rate is dependent on the size of the concentration gradient between a cell's exterior and its interior. Very small molecules such as water and oxygen and carbon dioxide move across membranes by simple or passive diffusion. Larger molecules, ions, and polar substances do not cross membrane by this method. Second is osmosis. It is the net movement of solvent molecules across a selectively permeable membrane from an area in which the solvent molecules are highly concentrated to an area of low concentration until equilibrium is reached. In living systems, the chief solvent is water. The three types of solutions which are normally found are isotonic, hypotonic, and hypertonic. Number 3, facilitated diffusion. The rate of diffusion across selectively permeable membrane is greatly increased by using carrier proteins, sometimes called permeases, which are embedded in the plasma membrane. Because the carrier aids the diffusion process, it is called as facilitated diffusion. Carrier proteins also resemble enzymes in their specificity for the substances to be transported. Each carrier is selective and will transport only closely related solutes. Because there is no energy input, molecules will continue to enter only as long as their concentration is greater on the outside. Two widespread major intrinsic protein channels in bacteria are aquaporins that transport water and glycerol facilitators which aid glycerol diffusion. The carrier protein complex spans the membrane. After the solute molecule binds to the outside, the carrier may change conformation and release the molecule on the cell interior. The carrier would subsequently change back to its original shape and be ready to pick up another molecule. The mechanism is driven by concentration gradients and therefore is reversible. Examples: glycerol is transported by facilitated diffusion in E. coli, Salmonella typhimurium, Pseudomonas, Bacillus and many other bacteria. This is prominent in eukaryotes where it is used to transport a variety of sugars and amino acids. Now let us discuss active transport. Active transport is the transport of solute molecules to higher concentrations or against a concentration gradient with the use of metabolic energy input. It resembles facilitated diffusion in the involvement of protein carrier activity, but differs in its use of metabolic energy and in its ability to concentrate substances. One example of active transport is binding protein transport systems or ATP binding cassette transporters, ABC transporters, are active in bacteria, archaea and eukaryotes. Now let us understand what ABC transporters are. Basically, these transporters consist of two hydrophobic membrane-spanning domains associated on their cytoplasmic surfaces with two nucleotide binding domains. The membrane-spanning domains form a pore in the membrane and the nucleotide binding domains bind and hydrolyze ATP to drive uptake. ABC transporters employ special substrate binding proteins which are located in the periplasmic space of gram-negative bacteria or are attached to membrane lipids on the external face of the gram-positive plasma membrane. These binding proteins bind the molecule to be transported and then interact with the membrane transport proteins to move the solute molecules inside the cell. E. coli transports a variety of sugars like arabinose, maltose, galactose and ribose and amino acids like glutamate, histidine, leucine by this mechanism. Bacteria also use proton gradients generated during electron transport to drive active transport. The lactose permease of E. coli transports a lactose molecule inward as a proton simultaneously enters the cell. Such linked transport of two substances in the same direction is called symport. E. coli also uses proton symport to take up amino acids and organic acids like succinate and malate. A proton gradient also can power active transport indirectly, often through the formation of a sodium ion gradient. In E. coli, sodium transport system pumps sodium outward in response to the inward movement of protons. Such linked transport in which the transported substances they move in opposite directions is called antipode. The sodium gradient generated by this proton antipode system then drives the uptake of sugars and amino acids. E. coli has at least a transport system for the sugar galactose. Now coming to group translocation. In active transport, solute molecules move across a membrane without modification. Many prokaryotes also take up molecules by group translocation, a process in which a molecule is transported into the cell while being chemically altered. For example, phosphoenol pyruvate sugar phosphotransferase system (PTS). It transports a variety of sugars while phosphorylating them using phosphoenol pyruvate (PEP) as the phosphate donor. Phosphoenol pyruvate plus sugar, that's outside. It gives pyruvate plus sugar phosphate inside. In E. coli and Salmonella typhimurium, it consists of two enzymes and a low molecular weight heat stable protein HPR. HPR and enzyme first are cytoplasmic. Enzyme second is more variable in structure and often composed of three subunits or domains. Enzyme second A is cytoplasmic and soluble. Enzyme second B also is hydrophilic but frequently attached to enzyme second C, a hydrophobic protein that is embedded in the membrane. A high-energy phosphate is transferred from phosphoenol pyruvate to enzyme second with the aid of enzyme first and HPR. Then a sugar molecule is phosphorylated as it is carried across the membrane by enzyme second. Enzyme second transports only specific sugars and varies with PTS, whereas enzyme first and HPR are common to all PTSs. PTSs are widely distributed in prokaryotes. Aerobic bacteria lack PTSs. Genera Escherichia, Salmonella, Staphylococcus and other facultative anaerobic bacteria have phosphotransferase systems. Some obligate anaerobic bacteria, Clostridium, also have PTSs. Many carbohydrates are transported by these systems. E. coli takes up glucose, fructose, mannitol, sucrose, N-acetylglucosamine, cellulose, and other carbohydrates by group translocation. Now coming to iron uptake. All microorganisms require iron for use in cytochromes and many enzymes. Iron uptake is made difficult by the extreme insolubility of ferric iron and its derivatives, which leave little free iron available for transport. Many bacteria and fungi have overcome this difficulty by secreting siderophores. These siderophores are low molecular weight molecules that are able to complex with ferric iron and supply it to the cell. These are either hydroxymates or phenolates catecholates. Ferrichrome is a hydroxymate produced by many fungi. Enterobactin is the catecholate formed by E. coli. Microorganisms secrete siderophores when little iron is available in the medium. Once the iron siderophore complex has reached the cell surface, it binds to a siderophore receptor protein. The iron is either released to enter the cell directly or the whole iron siderophore complex is transported inside by an ABC transporter. In E. coli, the siderophore receptor is in the outer membrane of the cell envelope. When the iron reaches the periplasmic space, it moves through the plasma membrane with the aid of the transporter. After the iron has entered the cell, it is reduced to the ferrous form. Iron is so crucial to microorganisms that many use more than one route of iron uptake to ensure an adequate supply. Coming to the last part of microbial nutrition, that is culture requirements of microbes. It includes culture media, isolation and cultivation of pure cultures. In the laboratory, bacteria are normally grown or cultured in either liquid medium, in flasks, bottles or large culture vessels called fermenters, or solid medium in petri dishes, which are round, normally plastic or glass dishes. Introduction of microbes into or onto these media is called inoculation. The nature of the medium depends on the microorganism's natural environment because its nutrient requirements reflect the natural surroundings. The culture medium must contain all the nutrients that the microorganism requires for growth, like water, a source of energy, carbon, nitrogen, essential inorganic ions, and a number of trace elements. Bacteria that can synthesize all they require from the basic ingredients are called phototrophs, and most microorganisms that survive in the outside environment can do this. Microbes that have become adapted to life in a situation rich with nutrients such as human body may require other growth factors like vitamins, amino acids or nitrogenous bases to be provided. These are called auxotrophs. The media that are used in microbiology laboratories to culture bacteria are referred to as artificial media or synthetic media because they do not occur naturally, rather they are prepared in the laboratory. There are a number of ways of categorizing the media. One way is to classify based on whether the exact contents of the media are known or not. Number 1. Chemically defined media. It is one in which all the ingredients are known and was prepared in the laboratory by adding a certain number of grams of each of the components: carbohydrates, amino acids, salts, etc. Particularly photolithoautotrophs such as cyanobacteria and eukaryotic algae, they can be grown on relatively simple media containing carbon dioxide as a carbon source, often added as sodium carbonate or bicarbonate, nitrate or ammonia as a nitrogen source, sulfate, phosphate, and a variety of minerals. Chemoorganoheterotrophs can be grown in a defined media with glucose as a carbon source and an ammonium salt as a nitrogen source. Number 2. Complex media. It contains undefined ingredients and the exact contents are not known. Complex medium may be sufficiently rich and complete to meet the nutritional requirements of many different microorganisms. They contain undefined components like peptones, meat extract, and yeast extract. Peptones are protein hydrolysates prepared by partial proteolytic digestion of meat, casein, soy meal, gelatin, and other protein sources like carbon, energy, and nitrogen. Beef extract contains aqueous extracts from lean beef and contain amino acids, peptides, nucleotides, organic acids, and minerals and vitamins. Yeast extract from brewer's yeast and contain an excellent source of B vitamins, nitrogen, and carbon compounds. 3 commonly used complex media are: Number 1. Nutrient broth. Peptone 5.0 grams per liter, beef extract 3.0 gram per liter. Number 2. Tryptic soya broth. Tryptone, enzyme digest of casein, peptone, enzyme digest of soybean meal, glucose, NaCl, and K2HPO4. Number 3. McConkie agar, pancreatic digest of gelatin, casein, peptic digest of animal tissue, bile salts mixture, NaCl, neutral red, crystal red, and agar. These media are routinely used for cultivation of bacteria in the laboratory and particularly useful for cultivation of bacteria whose growth requirements have not been defined. Culture media can also be categorized as liquid or solid. Liquid media, also called broths, are contained in tubes, flasks, and fermenters. Solid media are prepared by adding agar to liquid medium and then pouring the medium into tubes or petri dishes, where the media will solidify. Agar is a complex polysaccharide that is obtained from red marine algae. Other solidifying agents are gelatin and silica. 1% or 2% agar can be used to solidify, but 1.5% is the most commonly used. Enriched medium. It is a broth or solid medium containing a rich supply of special nutrients that promotes the growth of fastidious organisms that have complex nutrition and environmental requirements. It is usually prepared by adding extra nutrients to a medium called nutrient agar. Blood agar, nutrient agar plus 5% sheep red blood cells, it's bright red in color, and distinguishes between the hemolytic and non-hemolytic bacteria. Chocolate agar, nutrient agar plus powdered hemoglobin. It is brown in color and is considered more enriched than blood agar, as hemoglobin is more readily accessible in chocolate agar. Pathogens like Neisseria, gonorrhea and Haemophilus influenza, which will not grow on blood agar, can be cultured. Coming to types of media. Number 1. Selective media. These are designed to suppress the growth of unwanted bacteria and encourage the growth of desired microbes. Examples: McConkie agar inhibits growth of gram-positive bacteria and thus is selective for gram-negative bacteria. Phenyl ethyl alcohol PEA agar and colistin naldesic acid CAN agar are selective for gram-positive bacteria. Mannitol salt agar contains a concentration of 7.5% NaCl that is quite inhibitory to most human pathogens. Bile salts, a component of facus, inhibits most gram-positive bacteria while permitting many gram-negative rods to grow. This media is used for selecting intestinal pathogens, which contains bile salts. Dyes such as methylene blue and crystal violet also inhibit certain gram-positive bacteria like Staphylococcus can produce acid from mannitol and turn the phenol red dye to bright yellow. A medium containing acetate as a carbon source would be selective for organisms that grow on acetate. Sebarod's dextrose agar is used to isolate fungi and cellulose for cellulose digesting bacteria. Number 2. Differential media. Makes it easier to distinguish colonies of the desired organism from other colonies growing on the same plate. McConkie agar is also a differential and selective medium to distinguish between lactose-fermenting organisms, for example E. coli, and other non-fermenters, for example Shigella species. Lactose in the medium is fermented by E. coli producing acid, which causes an indicator dye to change color to red, and colonies that do not ferment lactose are white. Dyes can be used as differential agents because many of them are pH indicators that change color in response to the production of an acid or base. McConkie agar contains neutral red, a dye that is yellow when neutral and pink or red when acidic. Mannitol salt agar is used to screen for Staphylococcus aureus and it turns the originally pink medium to yellow due to its ability to ferment mannitol. In a sense, blood agar is also differential because it is used to determine the hemolytic and non-hemolytic bacteria. Blood agar is both differential and an enriched one. It distinguishes between hemolytic and non-hemolytic bacteria. Hemolytic bacteria, for example, many streptococci and staphylococci, produce clear zones around their colonies because of red blood cell destruction. With this, we conclude today's lecture. Hope you have understood it well. Thank you.